Hello friends, I am Dr. Anju Y Mundhe from Anna Sahib Magar Mahavidyalaya, Hadapsar, Zoology Department. Today we will discuss about the active and passive transport. Membrane permeability vary according to the nature, that is polar and non-polar, and the size of the molecule. Therefore, there are two determining factors, that is polarity of the molecule as well as the size. Now, the hydrophobic molecule like oxygen and carbon dioxide, they can easily cross the lipid bilayer, that is a plasma membrane. While the small, uncharged but the polar molecule like the water and the urea, they cannot cross the membrane easily. Some molecules are rejected, while the large, uncharged polar molecules like the glucose, because of its large size, only few molecules are able to cross the membrane and most of them are going to be rejected. In case of the ions, though they are very small in size, but because of its polarity, they are not able to cross the membrane. They are restricted. Passive transport. Passive transport, it is a movement of ions and the other atomic or the molecular substances across the cell membrane without need of energy or expenditure of energy. This movement of the molecule is along the concentration gradient that does not require ATP. Now this passive transport, it is of two types, simple diffusion and the facilitated diffusion. Now the first one, that is a simple diffusion. It is a movement of molecule from its higher concentration to its lower concentration up to the equilibrium. This process is comparatively slow. Example is oxygen or carbon dioxide. This is a plasma membrane. On one side, there is higher concentration of the one molecule is observed, and which is slowly and steadily it is going to be distributed on both the sides of the plasma membrane. Now the facilitated diffusion. It is a movement of molecules across the cell membrane via special carrier proteins that are embedded in the plasma membrane. Example, it is a transport of the glucose into the red blood corpuscle. It is also called as a uniporter because it carries only one molecule along only one concentration gradient from one side to the another side of the plasma membrane. This is extracellular environment from which the glucose molecule is going to be attached to this uniporter, and afterward the shape of the carrier protein is going to be changed and this molecule is going to be deposited into the cytoplasm through the carrier protein. Therefore, it is known as a facilitated diffusion. It is also along with the concentration gradient. Now, the active transport. Lipid bilayer are highly impermeable to most of the polar molecules, and because of which there is use of ATP is going to be observed to transport such a molecules. Active transport is always unidirectional. To transport these polar molecules, the sodium, example is sodium and potassium, the membrane proteins are going to be required. It means it is a channel or a carrier protein. It is against the concentration gradient. It means the polarity as well as the concentration gradient. These two are the driving forces which requires the ATP molecule to cross the membrane. Essential nutrients like amino acids, carbohydrates, ions are always transported inside the cell from the external environment, irrespective of concentration in the external environment. Now, this active transport, it is also of two types, primary active transport and secondary active transport. Now, the first one is primary active transport. Here, the active transport, it means it requires the utilization of energy. Shape of the carrier protein is changed by utilizing the energy of the ATP molecule. For example, sodium potassium pump. Here you can observe that this is a plasma membrane in which this carrier protein or the channel is present. Now, this is intracellular space, that is towards the cytoplasm. This is extracellular space. Now, because of its shape, from the intracellular, the sodium ions are going to be deposited in this channel, After utilizing the ATP molecule, the shape of the carrier protein is going to be changed, and these molecules, that is sodium, are going to be deposited into the extracellular environment. Afterward, the potassium ion, it is going to be deposited in a channel, and it is going to be transported towards the cytoplasm. Both these molecules are going to be transported against their concentration gradient. Therefore, there is requirement of the ATP to move the molecule against their concentration gradient. Now, the secondary active transport. By utilizing ATP, the sodium ion concentration gradient is going to be created in primary active transport. Now, the secondary active transport indirectly uses this energy. For example, sodium glucose co-transporter. Here, in this case, this is a plasma membrane. Here, A molecule is sodium ion and the B molecule is glucose molecule. If you observe that the A molecule, they are having higher concentration on one side against their another side. For example, sodium glucose co-transporter. Here you can observe that the A represents the sodium ion while B represents the glucose molecule. Here you can observe that with the help of the sodium potassium pump, that is primary active transport, we have created higher concentration of the sodium outside the cell and lower concentration inside the cell. Now, using this concentration gradient, the B molecule, that is glucose, it is going to be transported from its higher from its lower concentration towards the higher concentration against their concentration gradient. Now the SIM porter. If the membrane transporter moves two substances in the same direction, it is called as a SIM porter. Here you can observe that both A and B are going to be transported in one direction. Therefore, they are called as a SIM porter. While the antiporter, in contrast, the antiporters move the two substances in opposite direction across the membrane. Example is sodium hydrogen transport. Thank you.